Hey, let's get into transcription now. Transcription is the production of an RNA molecule that has the same information as the original DNA. And so DNA is the template for transcription. And so we use DNA and we make a copy of whatever the coding strand says by using the template strand of the DNA. So when you produce an RNA molecule, we typically have a region called the promoter site. A region called the promoter site, which is right here, which is where RNA polymerase binds. Frank? Sorry. They love to play. That's all I can say. So that's where RNA polymerase binds. And then RNA polymerase reads the DNA and produces an RNA transcript of that DNA. And it'll stop once we hit a terminator region, which basically stops transcription. So RNA reads the DNA and so there's two strands. We have a coding strand of DNA and then we have a template strand. Either strand can be the coding strand. So in this case, this strand up here is the coding strand, but it just as easily could have been this bottom strand could be the coding strand for another protein, okay? So in this case, this top strand of DNA is the coding strand. The coding strand is also known as the plus strand, whereas the template strand is known as the minus strand. We use this strand to make our DNA or our RNA. That's really bad. That says transcript, even though it looks bad. I was trying to keep it on the page. So this allows the RNA to have the same exact sequence as the DNA, except since RNA doesn't have thymine, anywhere that thymine was will have uracil. And so here's that plus strand, and here's that minus strand. We use the minus strand to make a copy of the plus strand. And if you look, see, G binds to the C, and G is up here. G, C binds to the G, and C is up here. The only difference is wherever we had a T up here, we have a U down here. You can go through this entire strand, and this RNA molecule is identical to that one. Okay, so these are copies, or that is a copy of the DNA. Okay? So the term monocistronic and polycistronic, sometimes when you're making an RNA transcript, you might only produce one gene. So this could copy and produce one gene. This could be one gene for, say, insulin. If this was polycistronic, you might have more than one gene. And in that case, hold on, I'm going to just erase the insulin. You might have some enzyme 1 and enzyme 2 that work together. That would be polycistronic. Monocistronic RNA is found in eukaryotes, whereas polycistronic DNA is found in your bacteria, so prokaryotes. So we typically produce just one gene for every messenger RNA, whereas bacteria produce a series of genes that work together for some function. And so here is transcription. We have this promoter site, that's where RNA polymerase binds. You can see that right here. RNA polymerase binds, and as RNA polymerase moves along the DNA strand, an RNA molecule is produced that is going to be identical to the coding strand of the DNA. It'll move until it hits a terminator sequence. At the terminator sequence, the RNA polymerase will dissociate, the DNA will wind back up, and the RNA molecule that was produced can then be translated or can be modified depending on what needs have to occur. So the promoter region of the DNA is going to determine which direction transcription occurs, and then will determine which of the DNA strands is considered the coding strand. Once an RNA polymerase moves past the promoter region, another RNA polymerase can bind, and so we can produce multiple copies of a single gene very rapidly. Hello. There we go. So let's look at transcription. Again, this is just my drawing of transcription. So you can see the coding strand is at the bottom and the template strand is at the top. So we have a promoter region where the DNA polymerase binds. A leader region is just a portion of the DNA that isn't actually translated. Oh, sorry. And then, so it is transcribed, but the leader won't be translated. And so here we would have the actual amino acids being coded for in the transcript right there. Okay. I'm going to stop right here and we'll get into translation in our next video. Have a great day. Bye.